Today, we're going to be covering genetic pest control, which I think is my favorite lecture of the semester. I think this stuff's crazy interesting, and it's kind of the direction that I think a lot of pest control is going to go in the future. So it's something that'll become probably more and more applicable as you move further in your careers. Just some announcements. We will be meeting in the alfalfa this week for lab. So if you remember, we were going to have an alfalfa IPM lab towards the beginning of the semester, and that didn't pan out because it was a little too cold for the pests. And they kind of planted over the alfalfa with soft grains. So there's not much alfalfa out there, but there's plenty of alfalfa around the edge. So I went out and swept some of that this weekend and turned up a bunch of interesting critters. So we'll go out there and sweep that, and it'll give us the same basic idea as if we had the whole field. It just won't be quite as good. But same drill as before. I will have a van. It will leave at about 11:50 to go up there. And the alfalfa field is at the corner of Bullard and Chestnut. Next week, we actually have a lab trip. We're going to Mulholland Orchards. It's out near Orange Cove, California. I will post an address with some directions on how to get there on Blackboard, and then in class we'll also go over how to get there, because it's just, it's pretty straightforward, but it's a little tricky when you actually get there, recognizing the actual drive. But they've got a biocontrol rearing facility. They raise parasitic wasps for biocontrol, and so it's kind of a cool operation. And the guy who runs it's pretty friendly. So again, I will have a van, but there's just one van, and there's only 11 seats in it. So if you want a seat in the van, you got to come and talk to me, and I'll make a list of people who I'm expecting to be riding along, and that way I can know when we hit that max and we don't have people showing up the day of without a ride.
After that, are we coming for the orchard one? Are we coming back here?
No.
Okay.
It's done after that. So we're going to go, we're going to tour, and then you're free to go.
If you want to get another van, I have my defensive driving. I have no problem driving.
You got defensive driving?
Yep.
All right. Let's talk after, and we might just get another van. Last year, when I did this, I found that generally, because it's the end of the day, a lot of people just wanted to drive themselves anyway, so they wouldn't have to ride all the way back to campus. So I wouldn't be surprised if we don't have a big interest, but it never hurts to get another person registered.
Are we usually timed back before 5 by chance?
For Mulholland?
Yeah.
Oh yeah.
Okay.
It takes about half an hour to get there, so it's kind of like be at Mulholland around 2:30, 2:45, and then the tour itself is anywhere between like 45 minutes and an hour. So we're usually done by 3:30 usually.
And nothing to do here before going.
No.
We just go.
We're just going to go. Yep. It's a sort of an obligation-free sort of thing. We'll just take a roll call at the beginning, and you'll get points for showing up. Any more questions about this? All right.
What do they rear at this rearing facility? Did I miss that part?
Parasitic wasps. They only rear Aphytis malinus, which is used for controlling California red scale. But it's kind of a neat thing. Basically, when you're rearing biocontrol facilities, what you're mostly doing is you're like a scale raising facility. You're just raising tons of scales for them to reproduce in. Well, the lab reports are graded, and it's a real grab bag, which is usually what the first lab report is. We had an average grade of an 81, the median was an 82, and generally what we saw was that some people did really well, and some groups didn't do so well. But as you can see, the minimum was a 66%, so nobody flat out failed, so that's good. Generally what I saw was that a lot of the points were just generally lost in formatting. So students who have written scientific papers before did better. People who are fresh to the format did worse. And I don't like to penalize people just because it's your first attempt, right? So my general policy is that what I'll do is I'll hand back the labs. I'll put them out at the end of class. You can collect them. You'll have 1 week. And during that week, what you can do is you can edit the papers based on the comments I wrote on it, and resubmit it, and then I'll regrade based off of that. Okay? So that's only for the first paper. The next 2 scientific papers you write, we won't have a regrade. But since this is the first one, it's handy to have a chance to rewrite since it's the first time. As far as main issues I saw were formatting, mostly around the introduction, results in discussion, a little bit of issues in citations. During lab today, we'll go over each of these in detail and give you an idea of what I was looking for and what I saw. Okay? So we won't touch that in detail now because we got a lot of stuff to cover today. So, genetic techniques and pest control. At the end of lecture last time, we talked very briefly about GMOs, and I mean very briefly. And the GMOs that we're most familiar with in pest control really are the transgenic plants, right? And so for those of you following along in the PowerPoint, I believe for this one, I posted it in its entirety. There's no blanks, so keep that in mind. So we focus mostly on the idea of how can we modify plants to make them resistant. So GMO plants tend to act like naturally resistant plant varieties as well. But recently, there's been a real shift in focus towards saying, why should we modify the plants when it's actually pretty easy to genetically modify the pest? We've got a lot of new transgenic tools that allow us to modify pests, insects, bacteria, fungi very quickly and easily. And so now people are interested in how do we modify the pest to provide control. So we're going to touch on 3 topics. We've got sterile insect technique, which is sort of the old-fashioned one, but it sort of sets up the future. We've got RIDL, which is the technique that they're using when they... who's heard of the transgenic mosquitoes that are being released in South America and parts of Florida? Sorry?
I just read it about it on the PowerPoint.
Oh yeah, that's great. There you go.
So yeah, I know about them.
So you know about it, right. So RIDL is the technique that's used for those transgenic mosquitoes. And then we've got gene drive. Anybody heard of gene drive? Oh, this will be fresh. Gene drive's really cool, kind of scary. So we'll see how it goes. So sterile insect technique. Sterile insect technique was really the first time that genetic modification of pests was used to provide control of them. And this was done way back in the 1950s and 60s. So the technique was pretty crude. We didn't have genetic modification techniques back then that really worked. In terms of we couldn't select a specific gene to modify, but we could modify the genes of organisms by blasting them with a ton of gamma radiation. Because that's one way to change DNA, admittedly a very sort of a scattered shotgun approach to it. But so as far as sterile insect technique is concerned, it's most easily explained in really the first pest that it was used against, which was a huge success story, which is this guy, the New World screwworm fly, which I'll just refer to as screwworm from here on out. So screwworm is a fly, Californiae, meaning it's a flesh fly, and it was a major parasite of cattle in the New World, typically in sort of the semi-tropical to tropical regions of North and South America. All right. So screwworm had this really sort of terrible life cycle that made it very problematic, especially to people raising livestock, which is that it's a parasite of warm-blooded organisms. So it flies around and it searches out for warm-blooded organisms that have some sort of wound on them. So basically the skin is broken, exposing some of the flesh. When a gravid female, so a female carrying fertilized eggs, finds one of these, she'll land on the wound and she'll lay a whole bunch of eggs. So here we have a wound, here we have a whole bunch of eggs laid on the outside of that. When the eggs hatch, the larva burrows into the wound and feeds on the flesh until it reaches its pupal stage, at which point it wriggles out, pupates in the soil, comes out as an adult, and then the adults mate and lay eggs all over, just repeating the cycle over and over and over again. So on the next slide, I have a somewhat graphic picture of a cow with wounds from screwworm, so that's your warning. If you're a little queasy about that, you don't have to look. But so, if left untreated, the screwworm could cause a lot of damage because the larva didn't just feed in the wound, they basically prevent it from healing, and they actually cause it to expand a little bit. So the wound gets bigger, and as the wound gets larger and possibly infected, it becomes more attractive to screwworms. So you get more screwworms laying their eggs on it, the wounds grow larger and larger and larger until eventually the host dies. In very severe cases, you would have a full-grown steer dying within a week of the initial infestation by a screwworm. So this could be a very serious problem. And in the 1930s, cattle populations in some of these southwestern states had grown so large that the screwworm populations exploded. And in states like Texas that were heavily reliant on cattle, they were losing almost 200,000 cattle every single year just to this fly species. So basically, they had this fly problem, and then around about 1945, the United States dropped 2 atomic bombs on Japan, and it ushered in the atomic era. And there was a lot of interest in how are we going to use atomic technologies, radiation-based technologies, not just for killing people, but ideally for the general good. And so a lot of really interesting projects came out of this. I was just listening to a podcast about radiation gardens, where people thought that what they would do is they would basically expose plants to radiation to try and develop mutations that could be turned into new crops. So they would grow these big circular fields, and right in the middle of the field they put like some sort of radioactive isotope, like a big lump of plutonium or something that would just irradiate the field, and they would come out and see if any of the crops had turned into something interesting. And it worked to a certain extent. Not real well, but I want to say, what is it called? I forget it. There's a very popular grapefruit variety grown throughout Texas and the Southwest that was produced by one of these radiation gardens. But anyway, there were lots of experiments like this, and people were trying them out on insects. And what they discovered was if you exposed insects to radiation at young life stages, most of them would die, but the ones that reached adulthood were generally sterilized. Their genes were just so completely scrambled up, they couldn't produce any sort of fertile offspring. And so what these researchers realized was that these irradiated flies could be used to control pest populations, especially in the case of these screwworms, because they had one aspect of their life cycle that made them really susceptible to this control, which is that female screwworms only mate once, right? They don't mate multiple times. They mate with one male, they collect the sperm, they use that sperm to fertilize their eggs. So if these females mate with a sterile male, then all of their eggs will be unfertilized. They won't be able to produce offspring. So what the researchers realized was that what they could do is that they could raise screwworms in a laboratory, and they could blast them with radiation at a young life stage. They could then rear them out to the adult stage, isolate the males, and then they could spread these sterile males over the area with the screwworms using aircraft. The idea is that once there are millions of sterile males just flooding the area, they will mate with the wild-type females, and all of the eggs that they produce will be sterile. They won't hatch. So females will come on, they'll lay their eggs on the wound, but nothing will come out of them. And so ultimately, if you just keep repeating this, and you keep releasing these sterile males, eventually you'll just drive the population of flies down, 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 until there's so few of them that they can't locate any wild males to mate with anymore, and you won't have any more screwworms around. And they ultimately figured out that you actually don't need to outnumber the wild males by much. You just need to have roughly 2 sterile males for every 1 wild male that's in the population. So essentially, use radiation to sterilize the males, release the males in the wild over and over and over again, and eventually you'll have so many sterile males that you've driven the population down to 0. So in the 1950s, they had sort of optimized this technology, and they started releasing these flies in the United States. And by about 1959, they had driven the fly completely out of the southeastern United States. By the mid-1960s, they had pushed it all the way down to Mexico, right? So just using this technology, they had completely eliminated this fly within about 8 years from the United States. Now, what I think was really great, was really clever of them, was they realized that to stop the fly from coming back, they would have to keep releasing flies along the Mexican border, basically forever, just to keep them out. Now, you could do that, but that's a lot of effort. That's a really big border. So what the USDA did instead was they teamed up with the Mexican government, and they said, we'll help you rear out these flies, we'll help pay for it, you pay half, we'll pay half, and we'll push these flies out of Mexico. And so over the years, 1980, 1982, they just kept pushing the fly. By the 1990s, they pushed it out of Mexico. Then they just traveled down Central America until they got to the Panama Canal in 2001. And at that point, instead of having to release the flies all along this Mexican border, they just had to control them along this little 20-mile section of the Panama Isthmus, right? So essentially, what they did was they set up a shop down there where they raised these flies en masse and just released them over and over and over again all year round to keep the flies out of North America. I don't know if they have any plans to push down into South America to eliminate them there. I think there's a little bit of infrastructure problems in that area. All right, but so just to give you some visualization, here we have in the 1950s where they were raising the flies on these big trays, putting them in these little target boxes for release, loading them up in airplanes, and they would just have some guy riding in the back throwing boxes out the window every once in a while as they flew over the target area. By the 1970s, they had really ramped this up, and they had gone from about 50 million flies a week to about 500 million every week. So they're really cranking them out. And right now, in Panama, what they're doing is they essentially have these great big tray racks. And you can see right here, here's one of these trays. You can see it's a little bit taller than an adult male in this case, although he might be a little guy, who knows. But it's a relatively tall stack. Each of these holds about 1.75 million flies. So we're looking at somewhere around, in all of these, 300 or 400 million flies in this room alone for release. So sterile insect technique was hugely successful in the screwworm, and it got a lot of popularity. People started applying it to other pest species. You can see it's been used against a lot of different fruit flies across the US as well as other countries, as well as a handful of lepidopteran pests such as codling moth, bollworm, gypsy moth, etc. And just a handful of other pest species that I've been working on. Now, there are some challenges, though, with sterile insect technique, because it doesn't work on everything. It worked really well on screwworm. It's worked relatively well on things like bollworm. But basically, screwworm has been sort of the ultimate success, and we haven't had anything near that close since.
We still get cases, actually, for the screwworm.
Of screwworm?
Yeah, there's one in Florida, I guess they found. They think it came from Haiti, and then I don't remember where they found one in Texas, too, but I don't know where that one came from.
Yeah, I would guess that they get introduced every once in a while from people shipping cattle from South America and the like. So I'm not surprised. Yeah. So actually, whenever medfly turns up in California, one of the control techniques they use is they release millions and millions of sterile male medflies as a control technique. I wonder if they do that with the screwworm as well. That if you find screwworm, they just douse the area with sterile males, trying to eliminate it as quick as possible. But so there are some problems, some areas where sterile insect technique doesn't apply. Primarily, the major challenge are that the insectaries are just really labor intensive. You produce both male and female flies. And so you have to spend a lot of time basically sorting male flies from female flies, which depending on the species can be relatively challenging or a lot of microscopy work. In addition, irradiation isn't really specific, right? And so you don't just sterilize the males, but you also oftentimes make them really weak because you may have cooked the genes that develop their muscles, maybe develop their honing skills, their ability to find females. And so the males you produce typically aren't quite as competitive for mating as the wild males. So you have to release a lot more of them than the wild males. Actually, when they first started this project, they had a bit of a failure on the Sanibel Island trial the first time they did it, because when they were collecting the males to breed, to create their... So basically they're keeping these male colonies, right, over time, and they're irradiating them. So they have to select certain males out in order to keep the breeding stock going. And so the researchers were collecting the male flies that were the easiest to catch. These were the least healthy and the weakest flying of the males. And so their colony just got weaker over time as they kept selecting the very weakest flyers to keep their colonies going. And so by the time they released these males, they basically were useless, weak sort of walking around males that couldn't find females to mate with anyway. So they had to restart from scratch and get a whole new colony going. And also, some insect life cycles just don't work with sterile insect techniques. Screwworm is great because they mate once, and if they mate with a sterile male, there's nothing, basically they're done. But there are lots of insects that mate multiple times. So they wouldn't just mate with, say, one screwworm, but maybe they'll mate with 3, 4, 5, a dozen screwworm males. And you only need one of those males to be fertile in order to produce fertile offspring. So it doesn't always work. Additionally, we've got things like aphids that reproduce asexually. Or we've got things like fungi and bacteria that have no sexual cycle whatsoever. So we can't even apply this technique to pathogens. So it works with some, but not with all pests. And that's where RIDL comes in. RIDL is a new technique that is really kind of designed to address some of these limitations and to create more of a targeted approach towards genetically modified pest insects. So RIDL, or RIDL, stands for Release of Insects with Dominant Lethal. And so what that basically means is that they genetically modify the pest insect so that the males are carrying 2 copies of a dominant lethal gene. So just as a review, most all insects are diploid, meaning they have 2 copies of every single chromosome, right? You get a copy of a chromosome from your mother, copy of a chromosome from your father, right? So homozygous insects have basically each of their chromosomes carries the exact same gene copy, right? They have the lethal version of it. A dominant gene, right, is one that if you inherit it, it's going to be expressed. You're going to see the phenotype no matter what the other gene you inherit is. So if you inherited a dominant lethal gene, it would kill you, regardless of whether you had 1 copy or 2 copies. So that's the basic idea with dominant lethal genes. And so basically, the difference here between RIDL and the fundamentals of RIDL versus sterile insect technique are similar. You modify the male insect so that they carry some sort of trait that kills the offspring. The difference here, though, is that the males are genetically engineered to carry this trait instead of blasting them with some sort of radiation. So basically, the trait they carry is very targeted to their species. So ultimately, the idea is that you have some sort of late-acting lethal gene that you've inserted into the male. You release the male into the population. It mates with the wild female. The female will produce eggs that produce larvae, but those larvae will die at some point before they reach their adult stage. In the case of the RIDL they're using against mosquitoes, it kills them before they pupate. So a general walkthrough. The insect is first genetically modified to have this lethal dominant gene. And the gene is what we call late-acting, meaning that it doesn't kill the carrier if they are in their adult stage, but it will kill their offspring. So basically, if you're an adult mosquito carrying this gene, you're fine. You're going to live a normal mosquito life. But if you reproduce, your offspring will have the gene, they'll die before they reach adulthood. The gene is also engineered to have an on-off switch. So basically, scientists will be able to turn off the gene so that it doesn't kill the offspring under very specific laboratory conditions. This allows the genetic engineers and the scientists to rear large numbers of mosquito in a lab, but when they release them and take them out of those controlled conditions, the gene turns on and kills the offspring. So basically, they rear these insects in mass. The males are released to mate with the females. The offspring receive a copy of the lethal gene, and then they die before they reach adulthood. And again, just like in sort of sterile insect technique, you release these males over and over and over again, and you basically suppress the population, And if you do this on a large enough scale, spread these mosquitoes all over the place, you can eventually wipe out the entire population. So eradicate them. So the advantages here, there's no irradiation, so the males are much more competitive than they would have been if they were irradiated. And it does not rely on females mating just one time. So again, basically you have a sterility gene that you insert, you mass rear the modified mosquito, you cut out the females and just release the males, and the idea is when the males mate, all the offspring will have that lethal gene and they will die. So where this is being used, the model really is an Aedes aegypti. So this is a major vector of a wide number of diseases in basically worldwide. It vectors yellow fever, dengue fever, chikungunya, and Zika virus. And what's really nice about this system, as far as releasing males are concerned, is that in mosquitoes, females are the only ones that bite. They need blood meals in order to produce their eggs, but otherwise they feed on sort of sugary food sources like fruits and nectars and things like that. Males don't ever bite. So you can release a ton of male mosquitoes into the ecosystem and it has no real impact on human health, which is really nice. Whereas if we were, say, releasing a whole bunch of male fruit flies into the system, it might upset people a little bit. Well, that's actually a bad example because male fruit flies don't damage fruit either.
But they're annoying.
They are annoying. Yeah, that's true. But yeah, so basically males are harmless, and so it's nice. You can release a ton of them. Another advantage is these guys are invasive. They're native to just one small part of Central Africa, and so if we were to, say, wipe them out from North or South America, nobody would really lose a whole lot of sleep over it, and we would be really unlikely to do mass ecological damage, because these species aren't originally from those habitats anyway. Oops, right. And so those are the basic advantages. People see this as a major pest, transmits a lot of diseases, you can release males en masse, and it doesn't really hurt anybody, and they are invasive. So it's a lot easier to make the argument for eliminating them if possible. So the one company that is really heading up this movement to release these modified male mosquitoes is an English company called Oxitec. They're really spearheading RIDL. Whenever you hear stories about how the Brazilian government is teaming up with a company to release these mosquitoes, they're talking about Oxitec, which I suppose almost sounds like one of those companies in like a Jurassic Park type movie, you know, where the hubris of scientists proves to be misplaced and they end up releasing something terrible. But don't let that shy you away from RIDL. But they have a relatively humorous, there we go. I enjoy their silly video about basically outlining how they do mosquito control. Oh no, they moved it.
Dengue fever is a flu-like illness and can cause intense joint pain in severe
for the season.
Central America is practically one of its worst.
Dengue fever, caused by a bite from an infected mosquito, it's known as breakbone fever. Symptoms include sickness and fever, extreme joint pain, and severe cases, bleeding from the eyes and nose. According to the World Health Organization, there are 50 to 100 million cases of dengue fever every year, and for thousands of people, it is fatal. Currently, there is no cure and no vaccine.
I contracted dengue from mosquitoes around my apartment. I had a lot of pain in my bones, particularly. It felt like someone was hammering at my bones constantly. I would wake up screaming in the night. The pain was so intense.
Originally from Africa, the dengue mosquito has invaded more than 100 countries in the last 50 years.
This is a jungle mosquito and a jungle virus that has moved to the urban jungle. And we helped it by by essentially transporting it all around the world.
There have been recent outbreaks in Brazil, Mexico, and the Caribbean, and every year it is spreading. The culprit for spreading dengue fever is the Aedes aegypti mosquito. We spoke to a male and a female. Aedes aegypti, thanks for agreeing to be filmed.
Not at all, darling. Come a little closer. I won't bite.
So, this is your home.
Yes, isn't it perfect, darling? I just moved from the plant pot in the hallway to be closer to my delicious family. Of course, Aid here doesn't partake. He's a veggie. Honestly, call yourself a mosquito. Well, Hades just follows me around. It's the only thing he's good at.
Have you heard of dengue fever?
Darling, I feed, I mate, I lay eggs. I'll bite you whether you're sick or not. If that means I pass your horrid little disease on to the next person, it's not my problem.
But you do realise that's why humans spray chemicals and fogs, because of you.
Darling, I know. It's all about me. Oh, you explain, Aid. I can't be bothered.
Well, humans have been using fogs to try and kill us for 50 years, and it does kill other insects. But we're getting used to it now. Besides, humans don't like fog in their own homes, so we're safe in here.
What about the Oxitec method?
What's that? Another useless attempt to get rid of us?
Take a look for yourself. In the UK, Oxitec scientists have found a new way to control the dengue mosquito.
The Oxitec solution is based on the fact that we have produced a strain of mosquitoes. Now, these ones carry specific DNA, so that when they mate with females, the offspring do not survive. So we release males into the environment. All of these males carry this DNA. They will seek out and find females, and there won't be any offspring. The female cannot tell the difference between an Oxitec mosquito and a wild one. So the more Oxitec mosquitoes we put into the environment, the more likely females will mate with them, and will bring the population right down. It's birth control for insects.
What do you think?
So you're going to release more mosquitoes to kill us off? Are you mad? That just means more bites, more disease.
But Oxitec mosquitoes are males, so they can't bite.
I'll be able to spot these Oxitec mosquitoes a mile off.
You won't, Aegypti, they're just like normal males.
Well, I will!
But you won't. To you, they will be the same as normal males.
Who do you think you are? You think humans can outwit me?
Humans don't do anything. Oxitec male mosquitoes do all the work. If they're released near you, the males will find you. As you said, it's what they're good at.
This is an outrage. Next you'll be telling me Aid is one of them. I've heard enough. Aid?
Dengue fever is a serious problem. 2.5 billion people are at risk.
Right, so it's decidedly silly, but I enjoy it. So that's the basic overview of the technology. Specifically what they're using in these Oxitec mosquitoes is they've encoded a lethal enzyme called TTAB. And this is basically just a gene regulating type of enzyme. And so whenever it's turned on, it produces so many gene regulators that it basically scrambles up all of the gene transcription in the mosquito and kills it. But they've got it designed so that TTAB only works whenever TET07 is turned on. So here we have the lethal gene, and then TET07 here is the regulator. It needs to be turned on for TTAB to be transcribed. So TET07 is turned on and off by the presence of tetracycline, which is a really common antibiotic. So essentially, as long as tetracycline is in the system, TET07 is turned off, TTAB is turned off. So what they do is, in the lab, they raise these mosquitoes in water baths with a little bit of tetracycline in them. And this allows them to rear them in mass. Because tetracycline is not widespread in the environment, once you release these males, their offspring will be raised in pools where there aren't antibiotics present, and so they will essentially have their gene turned on, the TTAV, and they will die. As far as field trials are concerned, in 2010, they did some early trials in the Cayman Islands, and they had about 80% population control. In 2014, tests in Brazil resulted in about 95% control within the cities where they were releasing these mosquitoes. And as I mentioned before, the Brazilian government has ordered more of these mosquitoes in order to get the Zika virus out under control. As far as I can tell, there's no data released on how effective this has been on the larger scale. So as far as RIDL in agriculture is concerned, there are some attempts right now in order to produce sort of RIDL pests, the Mediterranean fruit fly. So when Oxitec built this system for mosquitoes, they initially did proof of concept in medfly. So we know that there are medflies that can be released, that do have this ability. And they've done trials in the lab where they've had 100% population knockdown just by releasing these guys. My understanding is that there are no actual field trials. There was a team, this Leftwich et al., that was going to run some trials, but I haven't seen any results out of them. So I don't know if that's because they got cut or if it's because they didn't get any good results. Hard to say. In addition, there's work in RIDL pink bollworm, and I believe that pink bollworms, RIDL sterilized pink bollworms are the pink bollworms that are released in the Central Valley whenever they get pink bollworm infestations. So I think these are already being used to control them whenever they turn up. So benefits. Benefit of RIDL is that it's highly targeted, right? Generally, pest species only mate with their own species, so there's not much chance of these genes crossing over into another species. Benefit of RIDL is also that it can be shut down. Because the offspring are sterile, there's no chance that the gene will sort of grow in the population. You have to keep releasing males into the system in order to get this control. So if you see some negative side effects, you just stop releasing male mosquitoes or male pests or whatever, and it shuts the whole thing down. And thus far, it has been highly effective in the systems they've tried it in. Major challenges is that it only works if we're releasing the males all the time, like I said. So it's not sort of a one-stop shop. You can't release the males once and get control. You have to constantly be doing it over the entire season. Similarly, it doesn't work real well. Earlier I said it works well in systems where females mate multiple times. That was incorrect. This is the correct one. If the females are still mating multiple times, there's always the chance that they'll find a wild type and will reproduce with them. And again, it only works in sexual systems. And I think another challenge that I didn't put in here is that in this case, we are releasing genetically modified organisms into the environment. And that does have some ethical questions that need to be considered because we don't understand ecosystems nearly as well as we think we do. And so anytime you release something out there, you can't necessarily pull it back. With this technology, you can to a certain extent, but there's always the chance that something will happen we don't understand. All right, we got 10 minutes. We can get through this. So, RIDL came out in about 2010. It started getting really popularized in 2013, 14, 15. And already people are saying that's old hat. I was talking to a mosquito guy down at the UC Kearney research station, and he was saying that nobody's really interested in RIDL anymore. What they want to do is switch up to something a little more advanced. So ultimately what they're saying is, RIDL's great, it provides control, but you have to put a lot of effort into rearing mosquitoes, separating out the males, releasing them into the environment. And that's very time and cost intensive for a form of control. What if instead of releasing males all the time, we could release males once and they just spread the gene through the population over time, so that they're always providing that control over and over and over again. And so some people have figured out that a really useful way to do this would be that instead of putting a gene in that kills all the offspring, what if we put a gene in that only kills female offspring, so that all the male offspring survive, and you essentially have genetically modified males coming out of the eggs in the wild that will go out and mate with the wild-type females. So instead of releasing males every generation, you release males once, and all of their offspring are genetically modified as well. So in this example, right, you would have some sort of, say, female-specific promoter that turned on the flightless gene. So when the male mosquito that's genetically modified mates with the wild female, all of the males, their gene isn't turned on because they are male. But the females have this flightless gene turned on, and the females don't have any wings. So they can't go out and feed on people, right? So essentially, these guys are okay. They're still carrying around those sterility genes or those flightless genes. They can go out and mate with other wild-type females, but these wild-type females can't feed. They'll die out. They won't produce offspring. So there is one problem with this, and that is based on the fact that most pests are diploid. They have 2 copies of each chromosome. So essentially, in this first generation, with the dominant lethal carrying male, we've genetically modified this mosquito so it's carrying 1 copy of the lethal gene on each chromosome, so that all of its offspring get 1 copy of the lethal gene. The problem is, all of its offspring, right here, these guys, they're only going to have 1 copy, because they got the lethal gene from their father, but their mother didn't have it. So they only have 1 gene. So when they go out and mate with the population, so if we take this second generation modified male, right, we expect half of them to have the gene, to be this blue guy, but half of them half of their chromosomes are going to have it. I screwed that up. So to clarify, this is our genetically modified male. When he goes to mate, half of his chromosomes will have the gene, the other half won't. So when it mates with a wild-type female, we expect 1/2 of its offspring to be modified, but the other half won't. So ultimately, we're only going to be controlling 1/2 of all of its offspring, instead of controlling all of its offspring. Then, when these offspring go to mate, all of the offspring from the unmodified male will survive, and half of the offspring from the modified offspring will die. So basically, we're controlling 1/4 of the population instead of half. And then if we go down the generation again, we're controlling 1/8 of the population instead of that 1/4 or that 1/2 or all of the population. So as the generations pass on, we get less and less and less control, because only half the offspring will inherit the lethal gene every single time. So to solve this, scientists have come up with what they call a gene drive. And so a gene drive is essentially just any gene that spreads throughout the population at a rate that's greater than 50%. So essentially, if you have 1 copy of the gene, you're actually more than 50% likely to inherit it from a parent, if that makes sense. Right? And so essentially for RIDL, the idea is that you design a gene so that more than half the offspring will inherit the lethal gene, even if the parent only has 1 copy. So the way they do that is they use a system called a CRISPR mechanism. And you may have heard of CRISPR. It's in the news. It's becoming a really big deal right now. It's sort of a universal gene modification technology, where you can modify a gene in an organism down to the nucleotide for about $75. So inexpensive, can be used in most any organism. What they're doing in mosquitoes is essentially they're inserting a CRISPR with the lethal gene in it. And what a CRISPR does is basically a CRISPR is an enzyme that will copy the lethal gene over to the chromosome that doesn't have it. So basically CRISPR, let me start over with this real quick. So essentially, right, we've got a RIDL male that's got the modified gene in it. And inside its genetic code, it basically has 1 copy of the gene. Well, in its offspring will have 1 copy of the gene, and then a section that doesn't have a copy, right? One chromosome with, one chromosome without. What the CRISPR does is basically it cuts open the second chromosome, it copies the lethal gene, and it inserts it in there. So that you go from being heterozygous, having one copy of the gene, to being homozygous, having two copies of the gene, one on each chromosome. So essentially, all of the offspring you produce will have two copies. And that way, when they mate, instead of having sort of this system where half of the offspring end up with the transgenic trait, all of the offspring will. So that you end up with 100% control as we move through the generations. So, a recent paper, they tried this out in Anopheles gambiae, which is a malaria mosquito, and they found that by inserting one of these gene drives, they managed to get about between 87 and 99% success in copying the gene over to the other chromosome. So you could more or less guarantee that at least 9 out of 10 offspring will have two copies of the gene and will pass both copies on to their offspring. And they showed that just over 4 generations that the lethal trait in the population jumped from 50%, which was their initial rate when they infested the population. It grew to 70% within just 4 generations. And they predicted that essentially within 3 years, the lethal trait would hit fixation, that all the mosquitoes would carry this particular lethal trait, and you would wipe out the population. Alternatively, some people are saying, why do we need to kill mosquitoes? Mosquitoes in and of themselves don't cause people a whole lot of harm other than causing bites that itch. What if we can genetically modify the mosquitoes in a way that doesn't kill them, but actually makes them immune to the diseases that they spread to humans? So there's been some effort to basically genetically modify mosquitoes so that they carry a gene that makes it impossible for them to carry malaria, right? So if they were to feed on an organism that had a malaria parasite inside of it, the parasite would go into the mosquito, but it would die there. It would not be able to establish itself inside the mosquito. And so they're basically looking at ways to build gene drives into mosquitoes so that they can cause this trait to spread in the population, so that while we still have something like Anopheles gambiae around, it can't actually transmit any diseases. So we'll be okay without it. A team out of UC San Diego just released a paper where they showed that this was possible. They had success in establishing it in a caged population at 100% just through natural mating and a single release of these modified males. So gene drive is kind of the hot topic, but there are some major challenges to this technology, one of which is that pests can evolve resistance to it. That essentially all you need in a pest is some sort of gene that counteracts the effect of the lethal one. So if you put in a lethal gene that, say, stops the production of a certain enzyme in the insect that they need to reach adulthood, if the insect has sort of a mutation that allows them to override that blockage, then they'll still survive. The other major challenge is that gene drive, by its very nature, is designed to be perpetual in the population. Once you release it, it sort of spreads itself by itself. And so a major challenge to this technology is that once we release a gene, we can't bring it back. And so if this gene ends up having some sort of unintended consequence, or if it turns out that these insects can hybridize with other insects, or maybe the gene causes the mosquito to be slightly toxic to the organisms that feed on it or something like that. There's not really any way to bring it back. And so we just kind of have to live with those effects.
Well, wait, is there any evidence of those effects?
Not yet. So there's no real evidence to those effects. Admittedly, no one's released a gene drive into the wild. And so I think the basic idea is what we call the precautionary principle, which is that if there's a chance that it'll cause irreversible damage, then we should be exceedingly careful before we actually go and release it. So people aren't necessarily saying we're not willing to try gene drives, but people are saying, let's be really careful before we release gene drives. Let's not get too excited and start releasing genetically modified organisms that spread their own genes. in the wild until we know a little bit more about these systems. But so ultimately, what researchers are doing, though, is they're trying to figure out if they can build these gene drives with an on-off switch in it, so that they could release these, and then if there is a problem, they could flip the switch and turn it off. I'm hazy on the details of how they would do that, but there seems to be some optimism that it could be done. So on Thursday, we'll be shifting to biocontrol,